Dr. Ramchand.
Thank you and good morning to everybody. Special good morning to Professor G.M. Nair, Professor Jayakumaran Nair, and all the participants over this webinar. And as Professor G.M. Nair has mentioned, this was planned probably about a year back, and we both decided then, even if, as I mentioned, I thought I will give even in the COVID, but then it would have been impossible. I realized that. It's not easy because, you don't have sufficient energy to do that. Anyway, as Professor G.M. Nair has mentioned, our association goes proud several years, 40 plus years, and I used to call him Cheta earlier in the college days, which still continuing, but then we have also a lot of professional association and very interesting program we started with him, pair program, when he was Kerala Biotechnology Commission chairman. Subsequently, we have also had an opportunity to sign a kind of research collaboration with Inter-University Center for Genomics and Gene Technology. And also that I have connected, and he is also involved in several other industries for such wonderful program. And that program, what we started was an excellent program. And again, I am thankful to Kerala Academy of Sciences, Professor Jayakumaran Nair, and Inter-University Center for Gene Technology and Gene Technology of University of Kerala. I'm going to give the talk on discovery and development of phytopharmaceuticals with special reference to reverse pharmacology. I'm sure many people do not know how to translate the findings to make it a commercial product. Also on systems biology, nutrigenetics, nutrigenomics and epigenetics. If you really see, this is a very complex subject. Nobody will be able to give in 1 hour time. So I suggested to Professor G.M. Nair some time back, any time, and in fact Professor Christy Fernandez, Dr. Christy Fernandez, KSIDC also was very interested, and again do a workshop for 2 days, how the translation can be taken place, how to translate. Now if you see over here in these slides, you can see multiple companies, and these are all different companies, that Saksin Life Sciences, Theragen Biologics, Theragen Life Sciences, MagGenome, and suppose these are the companies where I am involved directly as CEO or CSO or as director, and various technologies are involved. Saksin is a biologics company. We are involved in developing anti-VEGF for macular degeneration. Theragen Biologics is another similar company involved in developing molecule for diabetic retinopathy. Theragen Life Sciences is an ANDA company, and MagGenome, what we have decided to put as part of the collaboration for this webinar. The reason is that one particular company, we need a lot of marketing. As the product development we have translated, and many of our COVID products are in the market now. So all the things were viral, molecular transfer medium, viral transfer medium, RNA extraction, rapid RNA extraction, RT-PCR. Yes, but on the other hand, other products which is related to MagGenome, which we have developed, DNA, RNA, protein, etc. Currently, last 8 months, nothing is being selling because no research is happening. Now, another one important thing is that the products, what I am going to talk is connected to SIPWISE technology, which is a Baroda-based company. They have sponsored a large research program for the next few years. Already 4 products we are going to enter into market, translated functional drinks. Now, what I am going to talk is that how you can translate your findings. Now, I have mentioned about phytopharmaceuticals, but phytopharmaceutical is a very general, generic term, but mostly I am going to talk on nutraceuticals. In India, nutraceuticals is a comparatively novel thing, new things, and food supplements. And I will be also mentioning to you how translation can be done, what is the regulatory landscape, and what exactly you can do. The reason I am telling is that all over India, all over the university, lot of work is going on in natural products. But just see how many products are translated. There are lot of extractions are going on. Like for example, curcumin or hydroxycitric acid. Many companies are involved in extraction. but really the value is very little. You will see that, I will be explaining what is the type of valuation, what is happening. The Indian nutraceuticals or supplement market, very poor. But on the other hand, the US, it is almost 50 to 100 times more. We are just 1 to 2% of what the US market, worldwide less than 1%. So what is the reason? Now, I am going to discuss with you, with my experience introducing product in the US market, how it is to be done, how you can translate. And the next 4 products what we developed here also is going to go. So this is in a very simple way I am trying to make you understand, and what is the difference between drug discovery, nutraceuticals or supplement discovery and development. So this is what I am going to tell you. Now, I have about 51 slides are there. Let me see that, and Professor G.M. Nair has already mentioned that I can take 1 hour. So maybe 1 hour, 1 hour, 15 minutes. And please do not worry about some of the slides where a lot of metabolic pathway details are given, and I will try to explain these things in a very simple way. Otherwise, the entire thing, as I mentioned, should go as a workshop. It will take at least 16 hours to 18 hours. Sometime we can do that. Now, the next 1 hour, the contents will be types of Biopharmaceuticals and how, or nutraceuticals. And in order to understand how the translation or translational research and the product development, you also need to understand the complexities of drug discovery. Because drug discovery, biologics discovery is different. On the other hand, nutraceuticals are different. Drug discovery is extremely complex, and comparatively, nutraceutical development is comparatively easy. And immediate translation, and you will be able to commercialize the product. Then I will briefly talk about nutraceuticals, phytopharmaceuticals, difference, similarity I will mention. And many people really may not be knowing much about how reverse pharmacology and how that approach can be used in developing products. The other thing is that genetics, genomics, epigenetics all has come in the last 10-15 years, especially single nucleotide polymorphism, the role of SNP in genetic variations, how the product what developed, whether medicine, how these are involved with nutrigenetics or nutrigenomics. So I'll be briefly mentioning about that. Then I will talk to you about some of the product and product development strategy. Now, what I want you to understand is that how quickly you can develop a product, put it into market. That's the most important thing. This is very, very rarely seen in India. Other than extraction, a lot of work is going on, publication, but very few products are translated. And you also need to understand, India is a place for Ayurvedic medicine. But why that is not sold much in the US or European market? There is a reason for that one. Some of the products are very good, but it is not sold at all, or very, very little is sold compared to nutraceuticals. Our sales of nutraceuticals and supplements are also less. On the other hand, if you see pharma, generic products, large number of products are being sold in the US, European market. So these differences you need to understand. And after this, you can ask any kind of question, I will be able to tell. Now, if you see about drug discovery, you know about NCS, new chemical entities. Now that's what everyone is doing, but India is totally focused on only generics. New chemical entities, now virtually nobody is working on that. If you see biologics, there are novel biologics. For example, let's say the vaccine what is being developed for COVID is called novel biologic or novel vaccine. But again, in the last 15 years, Indian biologics is totally focused on generic or biosimilars. What's the reason? Now, one more category of product is there called botanical drugs. Currently, these botanical drugs are there only in the US. It's a very small segment. Few years back, I heard that in India also botanical drug is going to come. Please do understand, botanical drugs and nutraceuticals are different. All these things are, there is a lot of differences there. NCs and biologics are clinically proven. There is a disease claim is there, nutraceutical, whereas it is not there. So this is another difference which you need to understand. If you are talking about the complexities of drug discovery, you can just see over here. Now, all of you know that very large number of human diseases are there. Currently, it is 35,000 human diseases, which consists of cardiovascular disease, probably about 3 or 4, and 3 or 4,000, and CNS base, about 11,000, cancers, about 3 or 4,000, and many other kinds of diseases are there. And really, if you go through the drugs which is developed, and please note that there is only 450 to 500 basic drugs has been developed for this 35,000 diseases. What are the things we have developed? Extremely good vaccines have been developed, anti-inflammatory, allergic, anti-infective. These are the only few areas. But realistically, many drugs for cancer now started coming. Cardiovascular, really we don't know where we are standing. CNS disease, schizophrenia, depression, mania, manic depression, Alzheimer's disease. So much work is going on. There is no drug is there. So this is the unfortunate situation. In this background, many people are interested for supplement research, nutraceutical, because it can be introduced, and some kind of therapeutic evidence, if it is there, the product can be a big product. Now, again, complexity of drug, if you are talking about NCE or biologics, you can just see the complexity. The first requirement is that validated biomedical hypothesis. What does that mean? You need to have a validated target. Target is what? Currently, everyone is working on 5 different types of protein, which is a receptor or an enzyme or a transduction protein or a transport protein. Those type of protein-based target you need to identify. Only after that you will be able to develop. And you have to go through all these various complex process. The drug has to be targeted. It cannot go to every organ and everywhere it can act. Very selective to the target of interest, that is protein target. It has to be very potent. You can see nowadays that the types of molecules what we are taking is extremely small. That means the potency. You can't take 1 gram or 5 gram or 10 gram of drugs. Even if there is no toxicity, it will not be successful. In the last 20 years, most drugs have come in micrograms to milligram. That's very important. The molecule or the API or the NC you developed has to be physically stable. You can't get hygroscopic and getting hydrolyzed. So it is very, very important. Now the most important thing is that a successful molecule has to be soluble in hydrophilic solvents. Otherwise what will happen? It will not be absorbed. It has to be permeable. Permeability, whether it is gastrointestinal tract or in the cellular targets, it has to pass the cell membrane. Then metabolically stable. What will happen? A drug which takes immediately and it is acted upon by cytochrome P450, the drug will be inactivated. It should be non-inducing. It should not induce any of the enzyme or receptor, other proteins, then it will become addictive. Reversible means after going and binding to the target, it has to come back to its original state, go back, metabolize and excrete out. Durable, because durability of a drug also is very important, because sometime what will happen, continuously you take a drug, it will become ineffective. Non-teratogenic, because after taking, you might be knowing about the story of thalidomide in the US. So it is very, very important now. Come from your diet, your food. What are the constituents of food? The food contains carbohydrate, complex carbohydrate, and also you have got cellulose type product which might have an energy but can't be hydrolyzed. Then you have proteins and amino acids. Then you have various types of lipid, whether it is phospholipid or fatty acid, etc., and sometimes unsaturated fatty acid. So these are the major component which gives energy, plus it contains all the vitamins, all the minerals. So these are the constituents, this is all what we require. Now if you see some other plant-based product, if you see about turmeric you are eating. Turmeric contains all these things, but the most important thing, it also contains something called curcuminoid. If you see about tomato, which contains lycopene. If you talk about gingerol. So these type of constituents are there in plant. So that also comes under the supplement or nutraceutical. But now, what I told earlier, the first thing, the major constituent which you are taking, which is compulsorily you need, that product. For example, if you purify an amino acid which is deficient in some kind of food, then it is called food supplement. If you see Horlicks, you will be able to see that they are adding lot of supplements. For example, fish oil they are adding. So that can be considered as, or Complan, because lot of additional vitamin D3, vitamin E, those type of things are there. Then that can be considered as supplement. So I just wanted some kind of understanding, kind of difference between dietary supplement, what is that? What is nutraceutical? Please do understand, interchangeably this is used, and definitely it is very difficult to explain, and various countries use various terms. That also you understand. Now the most important thing is that regulatory landscape worldwide. Now, one of the things I mentioned to you is that the US market is extremely high for nutraceutical. There is a reason. US FDA is the ultimate authority over there for controlling all the dietary supplements or nutraceutical product. And there is an act called the Dietary Supplement Health and Education Act of 1994. One more term is used, grandfathered product. Grandfathered product means pre-1994, worldwide, any of the supplements you have been using, that means nutraceuticals or food material, and it is extremely easy to introduce in the US market. This is something you need to remember. On the other hand, post-1994, if you are coming out with a new product, that needs a separate registration. So the difficulty comes post-1994, and if you are developing a totally novel nutraceutical which is hitherto unknown. So there is some differences there. Now, US FDA has kept absolutely easy to introduce this product in the US market, not like FSSA, multiple approval. You can just introduce, if it is a grandfather product, the only person who has to take the responsibility is that company which manufactures. That company will be able to straight away introduce in the US market by just giving a notification to FDA. And if they are in any doubt, they will come within 75 days, then you need to test and everything. But normally, normal, natural nutraceutical supplement is very easy, and this is one of the reasons in the US the market is very big, because introduction is very easy, but it is very different in India. There is few more reasons are there, but whatever I am telling, there is some changes, alterations are there, I will explain to you. And Europe, it is called EFSA. Let me tell you, it is much complex to introduce in the European market compared to US market, even compared to India. India, all these rules and regulations came very recently only. We never had a term nutraceutical, probably food supplements or those type of things, and this was probably under the control of ICMR, DCGA, etc. Now FSSA, Food Safety Standards Authority of India. Then Japan. Now the most difficult place to introduce product, two countries, Japan and Korea. There is some reasons are there. You have to be, it's not the product efficacy, product safety is very, very important. They are making sure that the way which you are extracting, and if you are using certain solvents, you can't. So there is a lot of complexities there to introduce in this thing. And Canada is almost like US, and FDA Canada is involved. Now, what exactly Dietary Supplement Health and Education Act of 1994? I will just read out. It's an act of the federal, the United States federal legislation, just like in Indian Parliament, and they have brought this rule in 1994. Basically, what they have brought is that in order to introduce supplement nutraceutical product in the US market, comparatively easy. Now, the reason is that the worldwide pharma market is 2,000 billion. And the US itself, almost 30% of that comes, probably 700, 800 billion. In India comes only 1.5%, so that means about 30 billion worldwide. Now US nutraceutical market comes almost like 80, 90 or 100 billion currently. It's like very large. In India, 1 to 2 billion, including our export. So this is the difference. And there is a reason why so much being sold in the US market. Now we are talking about dietary supplements or nutraceutical, which many times, I don't know, none of us have to take vitamins, supplements, because we have more than sufficient consumption is there. Second thing is that it is very difficult to produce a deficiency of vitamins because our intestinal flora produces. Sometime B12 you might have to be supplementary. Anyway, so few reasons are there. What is that reasons? Also in due course I will explain. So please, I will just read out this. The Dietary Supplement Health Education Act 1994 is a 1994 statute of United States federal legislation which defines and regulates dietary supplements. So there is a regulation is there. In case there is a problem, immediately FDA will come, and there are many cases. If they found there is some problem, adulteration, not developed properly, they will withdraw. Then manufacturers and distributors of dietary supplement and dietary ingredients are prohibited from marketing products that are adulterated or misbranded. You have to be very careful what you are putting in your label. That's very important. You can't tell the disease efficacy or disease claims at all. So it's quite important, that one. That means that these firms are responsible for evaluating the safety and labeling of their products before marketing to ensure that they meet all the requirements of DSHIA and FDA regulations. So what you need to understand is that, whichever is the company introducing the product, they will take responsibility, make sure that it is as per the guidelines which is given by FDA. What I am telling is that they will not control you unless you go away from the regulations. Now, the regulatory landscape between USA and in India, regulation for licensing and registration by United States Food and Drug Administration, USFDA, under DSHIA. In India, Food Safety and Standards Authority of India. And the definition is that USFDA defines nutraceutical as dietary supplements. This is what I mentioned. So there is only one category of product, dietary supplements. Everything comes under dietary supplement. On the other hand, FSSAI defines nutraceuticals as foods for special dietary requirement, for special dietary uses. For example, let's say Isabgol. You know that. Isabgol, very interestingly, in the US it is a botanical drug. In India it comes under nutraceutical. And what is a special dietary need? Some people who has heavy constipation, they can consume that one, and there is some efficacy is there. So that is that special for some dietary needs. Then Act, Regulatory Authority of Registration of Nutraceutical, Dietary Safety and Health Education Act, that is the Act, and in India it is Food Safety and Standards Authority of India. And in the US it is 1994, in India just very recently came. I think it's the FSSAI has probably settled now only with all the regulations. Then what they do is that, the role of USA in or DSHIA is that product licensing evidence, this is for the regulatory requirement for registration of a new nutraceutical. If you want to introduce a new nutraceutical, then things are different. By the way, there is one more term used called GRAS, generally recognized as safe. That's called GRAS. This is a term which is used in the US. Now, if you are going for a new product, in order to get it a GRAS status, so that there will not be any regulatory constraints, you have to go through all this process of product licensing, evidence, then requirements of safety and efficacy, labeling, health claims, manufacture under GMP, any adverse reactions, etc. Once all these are done, then once you submit, you can get the GRAS status. So that is the way it is done. Then after some time, that product, they will not come behind you. So this is the requirement for registration of a new novel nutraceutical. In India, it is product evaluations, licenses, health and label claims they check into, and there is different types of forms are used for development. GRAS is an acronym for the phrase generally recognized as safe. If any ingredients is not having a GRAS status, they have to go through the formal application status. That's what I was mentioning. Now, I'm just briefly given about the nutraceutical market, nutraceutical or supplements or medicinal food market worldwide. Now, if you really see, worldwide it is really growing in a very big way, and the growth is between 8 to 18% per year. In India, still it is very poor, between 2 to 3 billion currently per year. Please also note that 90% of the worldwide market is between US, Japan and Europe. 90%. The remaining countries, 10% only, that all other countries. Now the most important thing you have to remember is that why US has high opportunities sales as compared to other regions. That's because, as I mentioned, role of US FDA. Now US FDA, they control drugs, NCs, biologics, those type of things. But nutraceutical, they do not want to control too much, because what they thought, anyway it is comparatively safe, it is coming from food, let people use it, though not much evidence is there. Now these are the reasons. Easier filing and easy to launch a nutraceutical product in the US. Now grandfathered, the term grandfathered means, as I mentioned, pre-1994, any of the product that has been used worldwide is called grandfathered product. Grandfathered product, very easy to introduce. Grandfathered nutraceutical products exempted under DCR regulations. So you don't have to go through any kind of regulation. The acceptance and popularity of nutraceutical in general public is high. There is few reasons. In India, nutraceuticals or food supplement, doctors cannot prescribe. In the US also it is not prescribed, but please remember, most of the people are educated, they read. Now there is certain conditions set forth by the distributors to introduce a nutraceutical to become a very successful product in the US, which I will come. As I mentioned, there is no regulation, but on the other hand, these type of certain non-regulated requirements are there, because I am telling based on my own experience introducing few products in the US market. US government also promotes the use of nutraceutical as it helps to reduce individual medical cost and burden on the hospital. Now this is an unwritten thing. They will not publish why they want to regulate less. One of the reasons, that in the US, going to hospital, going to doctors is extremely expensive. And if people are worried about their health, for example, somebody has little pain here, there, absolutely not required to go to a doctor. Now, what currently people are doing is the small issues, whether it is gastrointestinal issue or their skin issues or their eye issues, they go to a nutraceutical, they read everything. Especially they need to know about the mechanism. How does it work? A clinical evidence is there. Tox study has been done. These type of things and publications are there. Then what they do is that directly go and buy things. Please also remember, the cost of nutraceuticals is far higher than pharma products. That also is there. Now, all what I mentioned is that, the regulatory US FDA or DCR requirement for introducing product. But now if you are going to US and if you want to be very successful, you have to go to a big company to distribute it, like Cargill, several billion-dollar company. There are about 8 or 10 big companies are there. Or you have to go to a distributor. What does distributor do? The distributor, if you are a formulated product or unformulated product, what they do is that they give to other manufacturers. There are 1,500 to 2,000 such distributors there. If you are getting into a very good distributor, your product can become big success. In order to be success, they ask for these requirements. As I mentioned, again, please remember, this is not a requirement by US FDA. The first thing they ask is that, is the product has a patent protection? Now, where a natural product can have a patent protection, because it is naturally, there is no product patent, but you can have a process or application patent, which is only available other than India, everywhere it is. Application means a particular constituent will be able to efficacy in certain conditions, like reducing cholesterol or triglyceride or something like that. So that type of patent protection. So patent protection, multiple ways it is done, and there is one interesting way is called the synergy composition, and that is the way we are filing the patent. Synergy composition is that when 2 or 3 components get together, how the metabolic pathway together, or how they can amplify the efficacy. So that is a synergy patent. Then toxicological profile of the nutraceutical. It is so important. Now everyone knows that none of this product has got a toxicological effect. But when you are formulating, they want to make sure, probably it's pharmacokinetics or improved absorption will be there. Does that produce any kind of toxicity? Normally, acute and a chronic toxicity will have to be done, but normally a set of toxicity studies are there normally for NCEs. Probably it will be about 50-60 studies are there. None of those things are required. If it's something novel, they will ask for Ames test. Those type of few early tests also will be there. Otherwise, simple toxicological evaluation, and preferably in a GLP condition. The other thing is that it is also important to show the clear mechanism. You can't just introduce something and tell. But then, for example, if you are talking about curcuminoid, everyone knows that curcumin works at anti-inflammatory pathway and many transcription process or lipoxygenase inhibition. So it is very, very important that your product has got a clear mechanism. Clinical evaluation, that's most important thing. Now in India, how nutraceutical clinical trials, the clarity is not there. So normally you get an ethics committee approval, is a general, normal, simple product. You can do a clinical evaluation. But please remember one thing, in the US, last 4 years, you are not allowed to do clinical evaluation of nutraceutical in patients. No, you can't do it in patients. For example, you tell that you have a product for brain function or enhancement memory. You can't use Alzheimer's people. You can use people, reasonably elderly people who can have a problem with little memory, which age-related problem. The same way, osteoarthritis. You are not allowed to use arthritic people. What you have to do is that you can select people and ask them a treadmill. There are some people will not have any problem for 30 minutes, 40 minutes, 50 minutes. On the other hand, pre-arthritis, you will be able to find out that 5 or 10 minutes time, they start paining. Such patients can be recruited. You cannot do the clinical trials in the US if you want to submit in people with diabetes. But pre-diabetics can be done. For example, 6.5 glycosylated hemoglobin level, less you can use it. Your fasting blood sugar 125, 130 you are allowed to do. For example, somebody who has got a 200 fasting and 8.5, 9.5 or no glycosylated hemoglobin, they become patient category. You can't do test on that. But in India still follows that one. That is not the right as per the DSHIA and the US FDA rules. And the other thing is that please remember, one of the most important thing is that pharmacokinetic profile. You may not be knowing that the major problem, efficacy problem with most nutraceuticals are very poor bioavailability. There are some products with bioavailability and some product has no bioavailability. Curcumin is an example. Unless you show that after taking that it goes inside in your bloodstream, it has no value otherwise. So it is very important to show pharmacokinetic profile. How this can be done? By improving the absorptivity by using the formulation. And most importantly, it should be devoid of solvents. You are allowed to extract with ethanol and methanol, but at the end of that, you should not have. There are certain conditions, 50 ppm to 500 ppm are allowed. But let's say that you are using chloroform, you are using acetone, or you are using some other solvents. Now, if you are telling the solvents may not be there, but if you are telling that this has been used in your flow chart, the product will not be taken. This is mainly in US, Europe and Korea and and Japan. Now all these should be devoid of heavy metals, or there are certain allowances are there. It should be free of aflatoxins. So these are some of the conditions to introduce in the US market. Now see, the nutraceutical, what are the key focus area? If you talk about drugs, a particular cancer or glucose lowering, cholesterol lowering, triglyceride lowering, these type of disease claims are there. That is called structure activity relationship. On the other hand, in nutraceutical, it is the system. For example, you can develop a product for cardiovascular health. Now you cannot tell that, the product you have developed is reducing, let's say, cholesterol. If that is there, that is a disease claim. I am not telling, you can see many people are doing that. But as per Indian law, as per US law, no. Cholesterol lowering will become a drug molecule. But you can tell this is for cardiovascular health. You are telling that bone, osteoarthritis, joint, pain can be reduced, that is a disease claim. So you can tell bone and joint health. The same way digestive health. You can't tell that it will immediately stop diarrhea. You can tell that you get for digestive health. Cognitive health. You can't tell that it is for, for Alzheimer's disease or senile dementia. No. You can't tell it is cognitive health, it is good. The same way metabolic disorders. For example, it is connected to metabolic disorders means, type 2 diabetes, obesity, hyperlipidemia, all those things, metabolic disorders. Sexual health, you can say. You can't say that, for example, Viagra and very clearly PDE5 inhibition. No. Then it becomes a drug, and nobody has ever developed such products in the natural product. But you can always tell this is good for sexual health. A lot of products are there in the market. Skin health, the same way weight management, immunity, improving your immunity, you can tell. Cancer prevention, protection, you can say. Then vision, improvements in vision. Then you can tell about women's health, men's health and liver health. These are the category of nutraceutical supplements which is available. These are the only product as such taken. Few more are also there. About 8 or 10 products are there. That means you purify to a single entity and which is used. Now the very first one which was used is Cyanocylin. You can see the structure. From salicylin, salicylic acid, which is a drug molecule, but then it is a derivatized, hydrolyzed from this. On the other hand, salicylin is the original molecule. And it is from willow of tree bark. It is for pain management. Cycloxin is related. Digitalin. That is mainly from foxglove plant. There are various type of foxglove plant is there. Very complex structure. First of all, such complex structure never will be able to synthesize also. And this is a drug. It has a drug status. Congestive heart diseases. When there is a problem with the heart, congestion, and the blood flow is affected. That's a very good molecule. Probably one of the earliest molecule. Artemisin from Artemisia plant. What is Artemisin? It is again used for malaria. The same way like quinine from cinchona plant. If you see, both these molecules, comparatively not very complex structure, may be able to synthesize, but this is two extracts what we have done. And our normal coffee, what we have done. Caffeine contains, caffeine is a drug, it has a drug status, probably 70, 80 years. You must have heard about APC, aspirin, phenacetin and caffeine, one of the earliest combination. Mainly it enhances metabolism and pain management. It is used. So these are the only few real drugs which is purified and used. On the other hand, you can see, go through the literature and see there is a connection between phytopharmaceuticals and drug molecule. Almost 40-50% originally came from plant. Now what is that? That is derivatized. Some simple structures can be derivatized because many of the plant product has a problem, instability even after extracting. Many times it is metabolized in the body, and the most importantly, it will not be absorbed. Now, considering all these things, you can take the molecule, take the pharmacophore structure. Basic structure is known. You can derivatize that. That's why these are all originally came from the plant. Many times, one more thing is there, most of the plant-based product, whatever is the reason, no one knows, because invariably less toxic. I'm not telling few are there, but mostly it is toxic. So one advantage is that, you will be successful in toxicity. But let me tell you, once you derivatize, you can't say, but most of the time it will be having much less toxicity. You can see a large number of products for, whether it is for chemotherapeutics, analgesic or psychoactive, these are all originally derived from plants. Even now people are trying to find out, but let me tell you, I don't think any antibiotics has come which has come from plant, but lot of people are working on that. That's a meaningless this thing, because it is very, very easy to synthesize an antibiotic from normal source, because when you are developing an antibiotic, you are targeting a bacterial protein, and you don't need to worry about your protein within the body as long as that do not interact. So it is comparatively, anybody going behind developing an antibiotic through plant is very tough thing. Now, you all have heard about this. This is the most famous nutraceutical molecule in the world. I have worked on curcumin and a bioavailable curcumin oil. Now, if you see over here, right side, an applications also has been given. Whether it is anti-inflammatory, antioxidant. Now, this is not a drug claim. You are not allowed to do that one. You can't say that, it inhibits it is anti-inflammatory, it can decrease the pain. Then it becomes a drug molecule. On the other hand, there is lot of in vitro evidences are there, some animal experimentation, but none of this is shown in clinical efficacy. What is clinical efficacy? Double-blind, placebo-controlled trial. You have to prove beyond doubt. Even if it is reaching, for example, in curcumin as an anti-inflammatory, it is coming somewhere near to brufen. That is fine, but so far nothing has been shown. Capsaicin, an extremely good molecule. We all know that. You take rasam and other things, immediate some efficacy will be there. You can see. But unless that is proved in a clinical setting, it will not have that drug efficacy. But then that's why these are all under the phyto-pharmaceutical, nutraceutical. Ginger oil. Ginger, especially emesis, emetic problem. But again, it doesn't have a drug status. Lycopene, anti-aging, antioxidant, anti-diabetic, which is from tomato. Then from resveratrol, from mundringa, grapes. From grapes it is obtained, again the same condition. Then diallyl sulfide from garlic, then indole 3-carbinol from cabbage, then sulfophane from broccoli. Yeah. So these are some of the things. So I just want to tell, none of this has got a drug status. And what I heard is that many of this cannot become a drug, because unless you show a complete clinical efficacy, but then very good nutraceutical. So it can be still a wonderful nutraceutical. And one of the product what I developed from many of these products I've developed from curcumin called Curculife is going very well, but then it has got a limited efficacy. That's what I wanted to tell. One more thing I want to tell is that one more category drug is there in the US that comes under botanical drugs. Now, what is the difference between botanical drug, nutraceutical and the drugs? That's only in the US, not in India, but they are planning to introduce that. There are two categories are there. In botanical drug, you don't need to purify. As long as you have an extract and you have a complete method for quality control, that is fine. So some of those products one introduced this, Cyanocylin. That trade name is Veregan. That's a botanical drug. It's not a highly purified one single component. It contains many things, and this is mainly used for certain wart, that means abnormal growth of this thing, and mainly in genital organs and other places. So this is a botanical drug. Then crofelemer, that's also a plant-based extract. That is a drug-induced anti-diarrheal product in HIV. HIV people, especially because of the effects of drug, This is inhibiting that, HIV mediated diarrhea. So this category is called botanical drug. Now in botanical drug, one more category is there. It can become OTC. OTC means over the counter. Cascara, that is our, I think, Cascara is a plant, Spanish-based plant, which is again used for constipation. The same way, psyllium, I think that is our isabgol, senna, sennosides. So these are some of the products in the US as botanical drugs, but OTC. No, in India it doesn't. You can go and get any of these things, but this does not come under botanical drug. So why these are called botanical drug? Because efficacy has been shown. There is a clinical efficacy has been shown. That's why it is coming under botanical drug. Now, current problems with the phyto-pharmaceutical formulation and development. Now, what are the current problems? Now, in the right hand, you can see there are hundreds and hundreds of papers are coming out. But how many are translated into nutraceutical or phytopharmaceutical product? Now, till now, very few research was happening probably 25, 30 years back. But a huge number of work is going on all over the world. But translation is not much. The reasons are the active constituents of herbal drugs are not well established many times. That is one of the reasons. Standardization, stability and quality control testing are relatively time consuming, tedious and highly priced. Now if you see about 5 products are there, 5 constituents are there. In the US, if you want to introduce all these 5 constituents, quality control should be maintained. You can't make the changes in the concentration. This is one of the reasons that, I always tell people don't go with a lot of, extract contain 15, 20 products because they will not accept that one. Your quality control will become very difficult. And you are not allowed to make changes continuously because people are worried about suing by consumer telling that, you have given earlier something and now you have given something. So it is very, very important that, standardization of your herbal extract to introduce in the US market. It's not followed. This is exactly the same thing is done in Ayurveda. Take the extract, but you have no idea about the quality control and the constituents. Herbal drugs are mixture of many constituents and hence difficult to extract. That is the other problem, because sometimes what will happen is that extract all the things will be very good, but many times extracting the entire thing is going to be very difficult. And if you want to have a very good quality control, 100% or 99.5% pure constituent, whether it is for LCMS, MS or HPLC, you require. Chemical variability of plant materials. Now, from India, if you talk about Kerala or if you talk about Kashmir or if you to Gujarat, and people have shown very clearly the constituent percent varies, and sometimes one constituent, what your interest is there, it will never be present in the other one. This is the major problem. So quality control. Influence of harvest, drying and storage is another thing, because the way which you are drying, way which you are storage also is very important. These are the major problem, and this is, if you want to introduce the product in the US, this all has to be addressed. I will very quickly tell about Professor G.M. Nair. Can I use another 15 more minutes? Hello?
Sir, please. Please.
Yeah, okay.
Yes,
Yeah. Now 15-20 more minutes. I will simply start about 11:15 only. So probably 1 hour 15 minutes. So probably 20 minutes I will finish. So I will go comparatively quickly. Probably you have all got some kind of idea. Now what exactly is reverse pharmacology? I will tell with some example. You see right hand side. In the allopathic medicine, there are a lot of reverse pharmacology examples are there. Aspirin originally long back introduced for the pain purpose. How much you have to consume is between 200 to 400 milligram, maybe 2 or 3 times, and that is used for cyclooxygenase or pain management, not inflammation, mainly pain, analgesic and antipyretic. That's why aspirin has been found. Now during the course in the last 30 years, people have found that this thins blood. Then what they have done is that this can be used for thinning the blood. Now they have found out in a clinical trial using 50 milligram to 75 milligram, most of the people, that blood thinning capacity is there. That means the clotting time significantly increase, and you know that aspirin is one more drug which has been asked to take everyone who is above 40 years, so that your clotting, or this will prevent any kind of cardiovascular events or stroke events. That's why. Now how it was done? Subsequently, they have found out aspirin at that dose has got a clinical efficacy, and the mechanism identified. The first mechanism is cyclooxygenase, or the inflammatory pathway. The second mechanism, they found out that in platelet there is a leukotriene. Leukotriene is one of the molecules. Leukotriene receptor is associated with the inflammatory pathway. This leukotriene is downregulated by aspirin, the synthesis of leukotriene. So it's a leukotriene inhibitor, and leukotriene is required for two platelets to aggregate together, or a clumping of platelets takes place through leukotriene. So this is a typical example of reverse pharmacology approach, and they studied the mechanism, everything, but the product was introduced and everything is not. The same way, EPA is a natural molecule, became a drug molecule. What was the evidence? Earlier, EPA has been given for fish oil, for multiple things, and during that clinical evaluation, they found out that EPA decreases triglyceride, especially those who have got 500 milligram or above, significantly reduces. Then that's the time Lavasa was developed, but then they studied the mechanism. Now that this is associated with an SNP for certain people, which has got very high level of triglycerides, and this reduces. EPA is known to reduce, and this is a typical example of Nutrigenetics, Nutrigenomics, and reverse pharmacology. So basically, you have to understand that EPA was introduced as a nutraceutical, which became a drug molecule. Everyone knows remdesivir. I also use. This is a reverse pharmacology approach, which is introduced last 2 months thing. Remdesivir originally for some other viral inhibition, and they have found that remdesivir inhibits in vitro COVID virus. Immediately within 2 months, 3 months, with very minimal clinical trial, because already existing drug, toxicity, everything is evaluated. Now US FDA approved to introduce in the market for COVID people in India also. And but unfortunately, I also took 5 injections and realized that very next day the WHO report has come that it has no efficacy. Now, but the idea is not whether it is efficacious or not. The product is introduced via the reverse pharmacology approach. What is the advantage? Remdesivir took 15 years to introduce in the market. This took 3 months, because we already know the safety and all the tox profile. The dosage regimen is known. You all know the chloroquine and hydroxychloroquine, and how Trump was, advocating the use of hydroxychloroquine. Again, that was a reverse pharmacology approach. But unfortunately, that those 2 molecules were COVID days, because suddenly they found out immediately some efficacy is found, and people know that this is not toxic. It is already been used for some other this thing, and it is introduced. But now I don't think anyone is using that one. Theoretically, it is available, but you can't use it for. Most people don't use it. Another very good example of reverse pharmacology in the allopathic side is chlorpromazine. Everyone knows that chlorpromazine is one of the best antipsychotic drug, but this is introduced in 1950s. Originally, it was an antiemetic. Subsequently, it has been used for anti-allergic. Then subsequently, it was used for antipsychotic. Then now one more application has come. It is for adenylate cyclase inhibitor, cyclic AMP synthesis for mucosal fluid transport. So multiple applications are there, and it's a very good molecule. And this is a very typical example of reverse pharmacology. So what you need to understand is that how reverse pharmacology, normally a drug to develop, you have to start from the mechanism, the target, then lead identification takes several years. In the reverse pharmacology approach, already marketed drugs, everything is studied, a totally different application. And this is what is called the alternate use or drug repurposing. So drug repurposing is done using reverse pharmacology, and sometime you might be able to use this for, if the Nutrigenetics and Nutrigenomics part, that also you can combine. But the most important thing is that just you have to understand what is reverse pharmacology and how reverse pharmacology can be used in our natural product work. If you really see, this is a little complex slide, but I will explain. Traditionally, you know that multiple extracts combined together, which is used for your whatever is the drugs or tablets or kashayam or lehiam, whatever it is. That is the traditional way of identifying extracting. Then, suppose in the course of these things you have identified as different use, or in the people are telling I have this problem but this is good for a second problem, then you know what you can do is that find out what is the content, whether that particular condition it is good or not. Now how it is done is that this extract can be used for a clinical evaluation, but it is very important to do a human clinical evaluation. Advantage is what? Because it is a natural product, and if it is a combination of many products, you can always do a clinical evaluation. Once that clinical evaluation is successful for a different condition, a natural product or Ayurvedic product which is otherwise used, you will be able to use it for additional development. Now what you need to do is that you have to do an activity guided isolation of active compounds. Suppose you know this is for, let's say, sugar control, or decreasing one of the pathways in the glycolysis or TCA cycle. Then what you can do is that you will be able to isolate using activity guided isolation process. Then one single component can be isolated. But in the meantime, this product can go through the process and introduce for that second application. But now, as a nutraceutical, this can be done comparatively very easy. But on the other hand, if you want to develop it as a drug, what you need to do is that isolate that particular molecule or group of molecules. Either you can introduce as a botanical drug or as a single molecule. But there is a lot of examples for. I have given EPA as an example, but not many examples is currently known, but people are working on that reverse pharmacology. But let me tell you, it's an extremely good methodology, those who are involved in developing the molecules. Now systems biology, again complex 3-4 slides are there. What I need to tell is that what exactly is systems biology. Now normally what we are doing is that when a drug is developed, the drug will be involved in the target and the mechanism which is involved in that pathway. That is the way drugs are developed. Once systems biology came, what is systems biology? If you see in our system, what are the things? One is the DNA or genomics, subsequently transcriptomics, proteomics, metabolomics. Now the entire system is connected with this one. Now these 4 things are involved in the controlling the whole thing, and it is interconnected. For example, we are talking about very isolated system, we are inhibiting, you can see how the other system changes, how this can be studied. Okay, I will just first read out this, what is written, then I will try to explain in much simpler way. Please do not worry about these complex slides because we don't have the time. Systems biology provides a platform for integrating multiple components and interactions underlying cell, organ and organism process in health and disease. So the study has to be highly integrated and connected and there is an interaction between all these things. For example, one metabolite will have effect on other metabolite too. Scope, understanding, prediction and control of human functional processes, all the function, whether it is disease or other things. Validation of drug target interactions. A drug interacts with a target, which are the other metabolic alterations can be done. Calibrate model systems for testing the drug for a disease. You will be able to make the complete system in silico, and you will be able to study, which is already available. Studying drug mechanism of action. These are the things, especially in metabolic disorders. Now you can see a complex slide again. Now left side you can see these are the systems, various systems, phenomics, the phenotype. For example, your color, your health, or these things, transcriptomics and proteomics, how the pathway is changing. Now you will be able to pinpoint this particular product is very good in such and such condition, because now most diseases, the pathways are elucidated, whether it is connected to CNS related. For example, schizophrenia, dopaminergic and GABAergic pathway. If it is connected to diabetes, insulin pathway. So the entire pathway you will be able to pinpoint, especially by alterations in the proteomic level. This is the way systems biology is studied. Then what basically you are going to do is that introducing this product of your this thing, which is of course bioavailable, how this is altering the system. Then you will be able to tell that if you study further, this particular product can be used for such and such condition which is involved in that particular pathway. This is basically the systems biology approach. So I mentioned briefly, what I have mentioned is about about how systems biology can be used for developing phytopharmaceuticals. So if someone wants to work on this one, what you need is that multiple types of extracts you require. You need multiple cell culture one, and you need to have a microarray methodology. Using microarray, how you will be able to see, and the proteomics methodology. Little complex, but this is some way of doing. The second possibility is that suppose you are doubting that particular mRNA, you can RT-PCR based, you will be able to do that. So these are some of the way, real-time PCR, you can do that. The same way, what is nutrigenetics, nutrigenomics and epigenetics? Now, we all know that we are all different. Why we are all different? Because of the genes. And what exactly is happening to the genes? It is not necessarily the gene expression process, because we all have an SNP, single nucleotide polymorphism. Now, many times some are unimportant, normally because many of the SNPs has been identified, but many more has to be identified. These SNP is involved in how the post-translational changes can be taking place. A simple example I will tell you. There is a protein called peroxisome proliferated activated receptor. So PPAR alpha, beta and gamma. PPAR alpha, I think, its gene is mutated, or there is an SNP associated with in the case of diabetes. 25% of the people who has got this mutation will get diabetes. The reason is what? Because this PPAR is involved in controlling multiple metabolic pathway. So if you identify that, you will be as a diagnostic thing. Now the second thing is that if you identify a nutritional sensor, what is nutritional sensor? Normally nutraceutical. Simple nutraceutical, because you know that during transcription translation process, the nutraceutical will have to interact, or nutritional sensors will have to interact with the gene. Now a particular nutraceutical interacts with that gene, especially that place, that can affect the transcription and translation process. Now this is the main reason that if you see, some people are extreme obese, but the food they consume is like anybody else. And there are some people you can see extremely lean, highly athletic. The reason is that I mean they eat also a lot, because the metabolism is different. These are all connected to nutrigenetics. We don't have much time to explain. Now nutrigenetics or mediated alteration can be controlled by nutrigenomics or nutritional sensors. These are all interconnected. Now, one more thing I want to explain, what is epigenetics? Because I'm not going to explain this slide, again little complex. What exactly is epigenetics is that your diet, your food is involved in again controlling the genes. You know, epigenetics is basically methylation or phosphorylation, those type of process. Now, epigenetics don't change your sequence, but the methylation, if methylation process or phosphorylation process changes, there is always an opportunity the transcription translation can be altered. A very typical example is, you might be knowing Professor M.S. Patel's study. M.S. Patel was from M.S. University, Baroda. He is a Professor Emeritus in the US. He has first gone through a study called pup in a cup using pups. What he has found is that in the first trimester, mothers who take extreme high quantity of like energy or carbohydrate or glucose or sugar material, these children are prone to become very obese. Now, this is one of the reasons in the US a very large number of obesity is seen, probably highly carbohydrate mediated food, especially first trimester. What is happening is that they identified that pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex is methylated over this course of time, and what will happen is that these children, mother will be fine, mother may not be will not be obese or anything, these children become very obese. Now what has happened, this particular genetic alteration, epigenetics, is already given. So I just wanted to mention these three things, which is nutrigenetics. What is that? Is your SNP alteration. Nutrigenomics is what? Your food can alter your transcription, translation and metabolic pathway. And what is epigenetics? Epigenetics, your food can alter your gene regulation through methylation process, and that will go for the subsequent generations. Now I'll briefly mention about the current product development strategy. This is some of the product which we are going to introduce in the next couple of months. Products are ready, already clinical evaluations have been done. Now if you see what we are developing is the product, one product right now for hangover, two products for sexual function, one for immune potentiation, one for cognitive function, etc. Now these products, what we want to do, take it to US market. If you want to take US market, as I mentioned, there should be a patent should be there. Now, as you know that I mentioned that if you are going for a new extract to develop, it is complex. Again, it will be like a drug product. We might never be able to develop also because it is coming from the natural this thing. So what we have done is that we have studied the entire products which is in the market and normally approved. For example, if you see in India, FSSAI approved about 160 or 170 products in the FSSAI. 160 products, I think. Now, as long as you use that one, you are safe. But how to get a patent? Because all these natural products is not easy. One of the easiest way is using, of course, you can develop a formulation, but it's a very what is that? Anybody can repeat that one. A process patent or a formulation patent. And there is something called application patent which is not available in India, but a particular application, and the product is unknown. So what we have done is that, that is the way we have done. We identified what is hangover. Hangover consists of probably people lose sleep after 2 or 3 hours, get up, and people feel very thirsty, people get headache, people whole body will pain, lose appetite, then feeling of vomiting. So these are the things. These are related to two different major conditions, but the inflammatory condition, which is pain and headache and inflammation, body pain, etc. The second thing is that connected to emesis or vomiting. So these two pathways we have studied. And we have studied, combined these two products, and the most important thing is that in this case, the combined efficacy should be there. That means the pathway which is affected by one product should be affected by this thing. Now something like this, 2 plus 2 becomes not 4, 2 plus 2 becomes 8. That means multitude of efficacy should be there. That is what is called the synergy patent. So this is what we have developed in cell culture studies, these two combinations, specific combination, and we will develop exactly the same way. And the drink is ready, the clinical evaluation is going on. Now, most important thing you have to remember in nutraceutical, even if you are successful or not, if you are successful in clinical evaluation, show very clearly that this is effective, great. If it is not there also, you can introduce. But then all these things should be there to introduce in the US market, but then Indian market, they are not asking anything. The other one is that, female sexual health. Many people have got problem associated with the libido enhancement. So what can be done? The pathways, again, there are many products in the market. Product means individual constituents approved or available. And we can see that, the hormonal changes, multiple hormonal changes, how this can be controlled. Then neurochemicals, the cyclic AMP, nitric oxide and cyclic AMP interactions. So how to control these things, again combine two different products and develop. The same way with male sexual health. But please remember, the male, one of the major problem for a period of several years is erectile dysfunction. Now, erectile dysfunction already have different types of molecules are available and very successfully introduced, and all over the world it is used. Now, if we are going ahead and developing something for erectile dysfunction, it will never ever be successful, because there is a very successful product is there. And if it is, of course, if it is as good as an existing drug, then it's a different thing. But even if it is like, but please remember, normally nutraceuticals will be having slightly better than placebo effect. But if it is significant improvements are there, it will become a drug molecule. So this is a second molecule we are doing. Another one is that